Hi everyone, I am Monkita and you are welcome to Ancosite. Today we are going to discuss about polymerase chain reaction or PCR and its practical applications. Polymerase chain reaction or PCR is one of the most widely used techniques in genetics which help researchers to amplify DNA sequence of choice from a very large genomic DNA. It amplifies the specific DNA sequence many folds in a very short period of time for detailed study. Invention of this technique by Carey Mullis and Michael Smith in the year 1983 bring about a giant revolution in the field of research, and for this invention they were awarded with Nobel Prize in the year 1993. Polymerase chain reaction can be conducted using very few ingredients to mimic in vivo DNA replication in vitro conditions. The components of PCR are number 1, a thermal cycler, number 2, isolated DNA samples, number 3, deoxynucleoside triphosphate or DNTPs, number 4, primer specific for target DNA, number 5, PCR buffer, number 6, Taq polymerase enzyme, and lastly PCR specific tubes. We will discuss about each components in following slides. The first component is thermal cycler. If we closely look, the name itself suggests its mechanism of action. If we divide the name thermal cycler into two parts, at first we have thermal, which is related to temperature, and then we have cycler, which refers to a machine that performs cyclic events. The main principle of PCR relied on thermal cycling. Repetitive heating and cooling for about 20 to 40 cycles, depending on the amount of amplified product needed, are carried out by thermal cycler. It contains grooves or wells to hold PCR specific tubes. Different thermal cycler machines have different number of wells, ranging from 25 wells to 384 wells, depending on the models of the machine. The thermal cycler shown here is a Biorad T100, which I have personally used, and it contains 96 wells. The next component is isolated DNA samples. Genomic DNA can be isolated from any organism by obtaining their tissue, blood sample, saliva sample, etc. We all know that nitrogen bases, namely adenine, guanine, cytosine and thymine, acts as the building block of DNA structure by forming hydrogen bonds. Here, when we conduct DNA replication in an in vitro situation, DNTPs or deoxynucleoside triphosphate acts as building blocks of newly formed DNA structure by forming phosphodiesterase bond in between phosphate group that are attached to the deoxynucleoside by the action of DNA polymerase enzyme. The most important ingredients required for PCR is targeted DNA specific primers. Now, what is a primer? Primer is a short nucleotide sequence that provides a starting point for DNA synthesis. In PCR, two primers complementary to the 3' end of targeted DNA sequence are required. When these primers bind to the complementary 3' end of the targeted DNA sequence, a small double-stranded structure has been formed, which enables the Taq polymerase enzyme to bind, synthesize and elongate new DNA strands. We should remember that designing of primers is the most crucial step in PCR protocol, and on proper designing of primers, the success rate of PCR is highly dependable. I will make a separate video on primer designing later on. Finally, we require polymerase enzyme along with suitable reaction buffer. In PCR, Taq polymerase enzyme is used, which is the thermostable form of DNA polymerase 1. This thermostable enzyme is used to withstand the heat required for DNA denaturation that is performed at the initial most step of the reaction. Not only Taq polymerase, but for proper functioning of all the components, reaction buffer is required. It generally contains magnesium chloride or any other bivalent ions. Now, let's look at the working principle of PCR. The main steps of PCR protocols are as follows. Number 1, initial heating. Number 2, denaturation. Number 3, annealing. Number 4, elongation. Number 5, final elongation. And lastly, final hold. Among all these steps, denaturation, annealing and elongation steps are the part of a repetitive cycle. The most initial stage of PCR is initial heating. This step is essential for heat activation of Taq polymerase enzyme. In this step, the thermocycler chamber is heated up to 94 degree to 96 degrees Celsius or sometimes 98 degrees Celsius as well, depending on the thermostability of the enzyme. This step lasts for about 1 to 10 minutes, and this step is a one-time step, as one-time activation of enzyme is enough for the entire protocol. The first step of cyclic events and the second step of the whole PCR process is denaturation. This step ensures the melting or denaturation of DNA under the influence of high temperatures ranging from 94 to 98 degrees Celsius. In this step, the hydrogen bond between the complementary bases of DNA that holds the double helix structure breaks down, yielding two single-stranded DNA molecules. This step actually mimics the in vivo function of DNA helicase enzyme. This step lasts for 20 to 45 seconds. The next crucial step is annealing. This step is essential for primer binding to each strand of denatured DNA. In this step, the temperature of the thermocycler chamber is reduced to about 50 to 65 degrees Celsius, which lasts for about 20 to 40 seconds. In this step, temperature plays an important role because efficiency and specificity of primers are largely affected by annealing temperature. I will make an elaborate and separate video on primer designing later on, where I will discuss about all these facts in details. After primer annealing, the next step of cyclic event is elongation. In this step, the temperature depends on DNA polymerase used. For PCR with Taq polymerase, the temperature for optimum activity lies in between 75 to 80 degrees Celsius, but commonly 72 degrees Celsius is used for this enzyme. In this step, Taq polymerase synthesize new complementary DNA template strand by using free DNTPs present in the reaction mixture. Here we have to note that Taq polymerase can only synthesize new DNA template strand in 5' to 3' directions and thus choose DNTPs complementary to that specific direction only. Taq polymerase condenses the 5' phosphate group of DNTPs to the 3' hydroxy group at the end of the nascent elongating DNA strand. It is a thumb rule that DNA polymerase can synthesize 1000 bases per minute. Thus, the time required for elongation step depends on the DNA polymerase and length of the DNA target regions that is to be amplified. At each elongation step, the number of DNA target sequences is doubled. With each successive cycle, the original template strands plus all newly generated strands become template strand for the next round of elongation, leading to an exponential amplification of specific DNA target region. With this step, the cyclic steps of PCR completes a single cycle. Multiple cycles are required to amplify target DNA to million copies. The final steps of PCR are final elongation and final hold. Final elongation step lasts for about 5 to 15 minutes at 70 to 74 degrees Celsius, which ensures elongation of any remaining single-stranded DNA molecule. The final hold cools the chamber to 4 to 15 degrees Celsius for indefinite period of time that ensures short-term storage of PCR products. Therefore, if we sum up the entire protocol, it can be said that all the ingredients, when loaded into the PCR tube and subsequently to the wells of thermal cycler, the first cycle comprises of denaturation, annealing and elongation give rise to hanging strand target DNAs. After third cycle, we started getting target DNA molecules. After nth cycle, approximately 2 to the power n number of copies of target DNA can be obtained. That's all for today, everyone. If you think the content of the video is really helpful for you, please do like my video and share it with other people so that they can also get benefited from it. And please hit the subscribe button along with the notification bell so that you get notified whenever I upload a new video on genetics. Till then, goodbye.